All right, so relating back to the properties from chapter 1 that we first learned about matter, we want to be able to figure out why gases behave this way, and why liquids behave this way, and why solids behave this way. And also, how come things are a solid, a liquid, or a gas under any given conditions. What it all boils down to is two things: the total energy that the particle contains, and the strength of the interactions between our given particle and anything around it. Okay? So when we're thinking about ideal gases, one of our assumptions was that they don't interact with each other. And that's not a very valid assumption, actually. But that is what the gas laws are based on. Okay? If we decide to include the strength of attraction or repulsion between particles, then we need to be thinking about what kinds of attractions and repulsions can happen. Those that happen between two different molecules or atoms or, two different particles are going to be called intermolecular forces. Okay? So intermolecular forces is the bottom picture. So we have one molecule, what is this, acetic acid, and another molecule of acetic acid. And there's a dotted line between them. This dotted line is to signify the attraction between the hydrogen on this acetic acid with the oxygen on this acetic acid. All right? All molecules have IMF, intermolecular forces. Everything does. The strength of those IMF are what determine whether the food dye mixes with the oil or the water. Okay? The food dye has very low intermolecular forces with the oil, but very high IMF with the water, so they mix. Okay? We've already studied intramolecular forces. That means within a molecule, so we called those bonds. Right? So there's basically three kinds of bonds: ionic, covalent, and metallic. Okay? There are four kinds of intermolecular forces. All of these two things together will dictate all of the properties of every piece of matter. So that's why we study it. So the weakest intermolecular force is called two things: either London dispersion forces, or a lot of the time, which, by the way, I'm going to abbreviate that as LDF quite frequently. Or the other synonym is called van der Waals forces. I'm going to write that here for you. These are just two different people who studied the same phenomenon, and so these names get used really interchangeably. Everything, every molecule, every atom, everything, every piece of matter has van der Waals forces. The way they work is you have one particle with a random arrangement of electrons next to another particle with a random arrangement of electrons. At some point in time, because electrons move, we're going to end up with a scenario where the electrons are kind of hanging out more densely on one side of that particle than the other. We call this a partial charge, okay? It's not like it's giving up any electrons or gaining any electrons. It's just that the electrons are all hanging out on this left side of the circle. And so we write a partial charge like this. It's a lowercase delta with a negative sign. If you have a negative partial charge, a negative density of electrons on one side of the molecule, then the other side is positive. Okay? So then when we look at the adjacent particle, what's going to happen is it's going to see this positive, partial positive charge, and the electrons that were randomly distributed in the adjacent atom are just going to suddenly be attracted to that. So we call this an induced dipole. Induced meaning it's not permanent, it's temporary, it goes away pretty quickly. It was caused by the random arrangement of electrons in an atom next to it. So everything has LDF, everything. That's like the default, the lowest energy thing, okay? The next highest interaction we'll have is called dipole-dipole. This is a permanent dipole, not an induced dipole. So when we talk about whether a molecule is polar or nonpolar, we're essentially asking, does it have a dipole? So if the answer is yes, it's polar, then we can imagine where the electrons are going to be more concentrated. And that is based on both the symmetry of the molecule, so we need to think about Vesper for that, as well as the bonds in the molecule. So here in this simple linear geometry, if we had the same two atoms on either end, it would be nonpolar, which would mean it would only have LDF. But here we have a hydrogen attached to a chlorine, and chlorine is far more electronegative than hydrogen is. So that means this bond is polar, which means, because this is a linear molecule, this whole molecule is polar. It in fact tells us that electrons are going to want to hang out on the chlorine side way more often than they want to hang out on the hydrogen side. We're going to make an arrow to show that one side is more positive and the other side is where the electrons are at. Okay? It doesn't mean they're always there. It just means that they are more often on that side of the molecule. We have a density, higher density of electron charge over there. Okay? So I like to make this plus sign at the beginning of my arrow, so I remember, oh, that's where the partial positive charge is, and that at the end of the arrow is the partial negative. Right? So now what this tells us, it's pretty interesting. Every time we have a sample of HCl, pure HCl, no water here, the dipoles are going to organize themselves so that the positive end of one molecule is oriented to the negative end of the other. That's because of that force of nature that we called electrostatic. In this case, that's an electrostatic attraction because opposites attract each other. Okay, so that's what dipole-dipole interactions are all about. I told you that everything has LDF, which is true, but that doesn't mean they have the same magnitude of attraction between molecules. So here we're going to look at the noble gases first in this purple chart. When we do that, we see that neon, which is the smallest of these noble gases, helium is smaller, but it's not on the graph. Neon is the smallest of these, and we see it has the lowest boiling point. Argon is next, and then krypton, and then xenon. And this is the same order that they are in group 18. It goes from bottom to top, from the top down. It's neon, argon, krypton, and xenon. It turns out the reason for that trend in boiling point is because as the atoms get larger, they will have more London dispersion forces. So that idea actually applies to everything, not just single atoms. So we can also look at diatomic elements like in group 17. Fluorine is the smallest, then chlorine, then bromine, and iodine. And we see the same pattern. Always when it gets bigger, the boiling point goes up. So then it's like, okay, maybe that's just for atoms. Maybe that's just for elements. So we can look at two different samples of organic molecules, which are nonpolar. So the only IMF they have is London dispersion forces. And so we can compare what we call normal pentane, which kind of looks like a caterpillar. It's a straight chain of carbons with hydrogens all around. And we can look at neopentane, which is the same chemical makeup, C5H12 for both of them. But as you can see, neopentane is more of a ball shape because the carbons are clustered toward the center. So if you wanted to see a Lewis dot structure without the hydrogens, that's what's going on with neopentane. And over here, that's what's going on with N-pentane, which stands for normal. When we compare these two things, the top one has a significantly higher surface area. It's more spread out. The bottom one is more contracted. So surface area is really the difference, right? And so that's true for these atoms and elements over here as well. The farther down the periodic table you go, the more surface area that atom has. So more surface area, a bigger size, in other words, corresponds with having an increased boiling point. Right? So N-pentane has a higher boiling point than neopentane, even though they're the same chemical makeup, because N-pentane has more surface area, a bigger volume. Okay? Shape matters a great deal when you're talking about intermolecular forces, but so does atomic weight. Both of these are factors here. We can compare atomic weight by looking at this chart, and we can compare shape by looking at these molecules. Dipole-dipole interactions are responsible for a lot of the way biology functions, because most biological systems are aqueous, right? So here we have a diagram of water, and you can see, I'm going to circle the partially negative charges in purple. And you can see this is just a regular diagram of liquid water. I will circle the partially positive charges in green. And they always match up to each other. A positive and a negative are always matched together. That's what this dotted line is showing. Okay? Not all dipole-dipole interactions are created equally. The more polar the bond is in the molecule, the stronger the dipole-dipole interaction will be. So this leads to actually a separate category of special dipole-dipole. We call them hydrogen bonds. And this is not a bond, okay? It's important to understand that a hydrogen bond is an intermolecular force. The distance is too big to be a bond, and it's too easy to break to be a bond. So IMFs are way lower in energy than proper covalent bonds are. So that's why we have dotted lines, right? And so this drawing here is a model of DNA, a little teeny piece of DNA. So here we have guanine on the left side of this pair. I'll circle it in green. And on the right side, we have cytosine. And so guanine and cytosine always pair up with each other. Ooh, this is Christmassy. I didn't do that on purpose. It's just luck. Guanine and cytosine always pair up with each other in DNA and in RNA. And that's because they each have 3 sites for hydrogen bonding to occur. We have to have one side that is a hydrogen bond donor. That's the one that has the hydrogen on it. And the other side is the hydrogen bond acceptor. Okay, so the acceptor has to be negatively charged. So I'm going to circle that in purple again. Not fully negative, right? So we're talking about partial negative charge. It's not like it has a minus on there. And our hydrogen bond donors are all NH bonds in this instance. You can also get hydrogen bond donation if you have an OH bond or an FH bond. One of my students one time said they remember that hydrogen bonds are fun. F-O-N. N. My captions aren't cooperating. Okay, anyway. F-O-N. Fluorine, oxygen, and nitrogen. The reason for that is they are the most electronegative atoms in the periodic table, right? And so if you connect a hydrogen to F, O, or N, it's going to be a very polar bond, which means it's really, really good at making a dipole-dipole interaction. Very polar. So I want to make sure you see this. This animation here is DNA. It's an actual X-ray crystal model. So for reals, not made up, not calculated. And what I want you to notice is as this spins, look right down the middle of the lattice here. And you'll see a gap right there in between every layer. That gap is where those hydrogen bonds occur. So it's important to understand that hydrogen bonding is not a bond, because if it were, our DNA would never be able to split apart to be replicated, and we would never have made it past one single cell. In fact, in my research in graduate school, it was based exclusively on the fact that if you take platinum and you put it in the middle of G and C, it will covalently bond them together with the platinum in between, and that ruins the DNA. Okay? So hydrogen bonds, even though they have the word bond in it, are not bonds. Don't tell me they are. I might cry. Okay? Hydrogen bonds are more temporary than actual covalent bonds. They are longer, and they are lower in energy. They are, however, absolutely critical to the way that solutions are made, the way that biology happens, the way that everything in the world occurs.